All right, so we are going to go over a few problems on the cytoskeleton activity that was assigned for Chapter 17. I had you guys submit a survey and indicate what questions you wanted to go over, so I'm going to pick a few here. Just scrolling through this worksheet, I want to emphasize that on question 1, that you really take your time on this. As you read through, as you read through the chapter, they talked about all these different cytoskeletal components and where they can be found. So the answer to this is not going to be found in your book in one spot. You're going to have to really scan through the chapter or Google these different structures or processes and see what you can find, all right? For most of them, there's only one answer. For a few of them, I will allow more than one. So the first one that I want to go over is question number 4. Intermediate filaments allow sheets of cells to stretch out without breaking, right? So what do the intermediate filaments bind to? I don't know how much they talk about this in Chapter 17, but we've talked about this idea a lot in the past, how they are spanning the length of the cell and they are binding at these cell-cell adhesion points, all right? And if you have watched or when you watch Chapter 18's lecture, you'll find that those cell-cell junctions are called desmosomes, all right? So I will accept cell-cell junctions. I will also accept desmosomes or hemidesmosomes as well. Now, what structural property allows these filaments to stretch? To answer this question, you're going to need to look in your book at the section on intermediate filaments where they describe how these are, what their structure is like. All right, so I've got some, these are just images from the text that I have on file. And so in this figure 17.4, we can see intermediate filaments and how they are structured. The most basic unit is a monomer, this polypeptide, and it is an alpha helix in structure, and it will pair with another of the same protein to form a coiled dimer. Then these dimers assemble into tetramers and so forth until you get this big bundle, this filament of basically composed of these coiled dimers. Now, again, we've talked about how these intermediate filaments have a little bit of stretch compared to microtubules or actin. Microtubules and actin, they have a lot of little pieces like Legos, right? And you can imagine that those can snap. They can also, in terms of changing their length, the way they do that is not by stretching, but by removing monomers or adding subunits. And so in the case of intermediate filaments, they don't really attach or detach very well. See how staggered everything is? It's not really built to do that. But it does remind you perhaps of rope, right? How rope is kind of made of twisted fibers. And so rope is really strong, but it does have a little bit of stretch to it. And so because of this twist in the dimer shape, that gives it a little bit of flexibility. And the fact that this entire filament is made out of these coiled dimers allows that little bit of give, all right? So the coiled dimer structure, a coil of two coils, allows for a little bit of stretch in the intermediate filament. The next question that I want to talk about is, I guess we can go over number 9 if you like. Some people requested that one. The hydrolysis of GTP to GDP carried out by tubulin molecules does what? All right, and so we've got these different options here. So to answer this question and number 10, we're going to look closer at this figure. All right, and so there's more to this figure if you look in your book. Let's see, slide 47. Also, there are some excellent videos online, animations of this that I encourage you to watch if you haven't searched or if I haven't shown you them in the past. If you look up microtubule or actin polymerization, there's some great animations because, again, anytime you're studying something that is involving movement, it really helps to watch videos or animations showing that. So what we have here is a growing microtubule and a shrinking microtubule. When we say that the cytoskeleton is dynamic, we mean it can change in length. It can be shorter or longer. And so polymerization is what we're talking about with elongation, adding polymers or subunits. And depolymerization is when it's shrinking in length, it's losing those polymers. And so when we're talking about microtubules, the subunits that make up a microtubule are called tubulin dimers. And they are going to only add onto one end of the microtubule. When they attach, they have GTP bound to them, right? And we've talked about GTP-ACE proteins before. These proteins, they kind of inactivate themselves. They hydrolyze that GTP to GDP. It loses a phosphate, right? And so in this case, when these tubulin dimers have GTP attached to them, that stabilizes their association in the microtubule. So that helps them to stay, all right? And they are going to form basically this cap. And the reason why the entire microtubule is not all GTP attached is that, as we said, with time, eventually this tubulin protein is going to hydrolyze the GTP, and now they're going to be, instead of being bound to GTP, it's a GDP molecule, right? And so with time, they're switching over, and so that's why the back half of this microtubule is GDP bound, and only the top end, the end that's being added onto, the molecules that have been added most recently, are going to be bound to GTP. Now, if this slows, if, for example, the cell runs out of GTP-bound tubulin dimers, for example, eventually all of those molecules in that cap are going to be converted to proteins that have GDP. And so GDP protubulins don't associate as well, right? And so if they don't have this cap on here, this is what helps them to stay in this microtubule. If that cap is lost, they will fall apart. They kind of peel away and it depolymerizes and the microtubule shrinks, right? And so that's why this is really good to watch in video form because you can kind of see this dynamic process. Right, so this GTP bound tubulin is going to associate better with the microtubule, and if you have enough of it for a cap, even though the rest of the tubulins that are bound to GDP don't associate as well, they're not likely to escape or fall apart when this cap is present. Only when the cap goes away. And so how does this cap go away? It's when these proteins that have been recently added, after a certain amount of time, they're going to lose that phosphate group and be bound to GDP, just like the rest of these. And so if you're not continuously adding new GTP-bound tubulin, you will lose that cap. And when that cap is gone, now these GDP-bound tubulin that are unstable are free to fall away, right? So let's go back to our worksheet, right? The hydrolysis of GTP to GDP does what? Does it provide the energy needed for the tubulin to polymerize? No, because it's not that the GTP will only bind once it's converted to GDP, right? It's not like it needs energy to polymerize. They polymerize and then later they lose that phosphate group. Question 2 doesn't really make sense, all right? Hydrolysis of GTP to GDP, that does not promote assembly. In fact, if anything, it promotes disassembly. And so the ability of microtubules to grow and then to shorten and to grow and to shorten, this is called dynamic instability. And the reason that it will fall apart is because GTP gets converted to GDP, all right? So the correct answer for 9 is going to be D. But make sure you understand why. Right? So when we talk about question 10, we've got this GTP cap that stabilizes the end of the microtubule. And if the addition of these dimers, these GTP-bound dimers slows, how will the composition of the microtubule change, right? So these tubulin dimers that have been attached, over time, they are going to hydrolyze their GTP molecules. They're going to lose a phosphate, and now they're GDP. So over time, if this addition slows, eventually the entire microtubule will be formed of tubulin that is bound to GDP. Okay? Will this promote stability or destabilize the microtubule? Remember that this cap is important for stabilizing the microtubule. And so if that cap doesn't have GTP in it anymore, it's not going to stabilize the molecule. So that will promote destabilization. And will that promote lengthening or shortening? Shortening of the microtubule filament. The last question that I want to go over is number 22. This is another one that if you can find an animation for it might be helpful. Calcium binds to troponin and slightly changes the protein's conformation. How can increased calcium concentrations lead to muscle contraction? And make sure to describe the role of tropomyosin in your answer. I was not a big fan of the picture in your book, so I found one online that I liked a little more. Right? It's a bit fuzzy, but I think it is a little bit more detailed. So in this picture, in A, we have a relaxed muscle, and B is contracting. We have actin shown in blue, tropomyosin, and troponin. Now in B, when the muscle is contracting, what is causing this contraction? Right? What is binding to what? We know we have actin that is getting pulled, right? What is it getting pulled by? It's getting pulled by myosin. Now myosin is not shown in this picture, but they do show where the myosin attaches, right, on these black spots. So that is the region of the actin that the myosin has to be able to bind to in order to contract. So if these myosin binding sites are not available, the muscle cannot contract. Now, when the muscle is in the resting state, those binding sites are covered up by tropomyosin. So myosin could not, even if it wanted to, it could not attach and pull on the actin filament. When calcium is increased in the cell, so the cell receives a signal that it needs to contract, calcium is going to rise in that muscle cell. The calcium is going to bind to troponin. When calcium binds to troponin, it slightly changes its conformation. You remember that structure determines function. So if troponin is associated with tropomyosin, if troponin moves, it's going to also move tropomyosin. And the movement of tropomyosin exposes the actin so that the myosin binding sites are now available. So now the myosin can interact with the actin and pull on it. All right. So you got to make sure that you talk about calcium, troponin, tropomyosin, actin, and myosin. And again, there are animations out there that can help demonstrate this dynamic process as well. Before I go, I want to note that for question 23, pay very close attention to detail, all right? There's a figure very similar to this in the book. There's also animations online, and if we were in class, I would watch an animation with you, but you are going to have to do a little Googling on your own. But make sure that you're really careful about what's happening during ATP hydrolysis and what's happening, how does the power stroke occur, all right? If you have any other questions about this assignment before you submit it, feel free to contact me. Otherwise, I will be seeing you around.